Welcome to this lecture on insect vectors and diseases. Today, we are going to explore a fascinating and critical topic, the host-vector relationship. If you have ever wondered why certain mosquitoes seem to target humans while others ignore us completely, you are in the right place. Understanding this relationship is the very first step in preventing deadly outbreaks of diseases like malaria and dengue. Let us dive in and uncover how these tiny insects make such impactful decisions. To start, let us imagine a food delivery app. In this scenario, the driver is an insect, the restaurant's food is a disease-causing pathogen, and you are the customer. For you to get the food, the driver must know where you live and successfully hand over the package. In nature, this exact scenario plays out every single day. The insect is the delivery driver, which we call the vector. The human or animal is the customer, known as the host. The host-vector relationship is simply the study of how, when, and why these insects find and bite their specific targets. Now that we understand the basic roles of the host and the vector, let us look at how insects choose their targets. In medical entomology, we study a trait called host preference. Some insects strictly prefer to feed on humans. We call this trait anthropophily. Other insects prefer to feed on animals, like cows or birds, which is known as zoophily. Why do we care about this? Because if a mosquito species only bites birds, it will never transmit a disease to humans. It is completely harmless to us. But an anthropophilic mosquito is a highly efficient disease transmission machine. Beyond just who they bite, we must also consider where they bite. This brings us to feeding behavior. Some vectors prefer to hunt and bite inside our homes. This is called endophagic behavior. Others prefer to stay outside, which is known as exophagic behavior. Understanding these highly specific behavioral traits allows scientists to map out exactly how a disease moves through a population. For example, if we know a mosquito is endophagic, public health officials can design targeted control strategies, like distributing insecticide-treated bed nets. Without this knowledge, we would waste resources spraying chemicals in the wrong places. Since we know that preferring humans makes a vector dangerous, we need a way to measure this mathematically. This brings us to the human blood index formula. Conceptually, this formula calculates the percentage of times an insect chooses to bite a human compared to all its available choices. In the equation, HBI stands for the human blood index. NH represents the number of insect blood meals specifically identified as human blood through laboratory testing. NT represents the total number of blood meals analyzed from that insect population, regardless of the animal source. If the value of NH increases while NT stays the same, the human blood index increases, meaning the insect is becoming much more dangerous to humans. Let us see this formula in action with a concrete example. Suppose a team of entomologists collects 500 blood-fed mosquitoes from a village. After running DNA tests, they find that 350 of them contain human blood. We are asked to calculate the human blood index. We choose our formula because it perfectly isolates the human preference ratio. We set up a simple division problem, placing 350 as the numerator and 500 as the denominator. Dividing these gives us 0.70. This final answer means that 70% of the time, this specific mosquito chooses to bite a human. It is highly anthropophilic and incredibly dangerous. Now that we have calculated the human blood index, We must realize that preference alone does not tell the whole story. Even if a mosquito loves human blood, it might only bite once a month, or there might only be 2 mosquitoes in the whole village. To truly understand the risk, we measure the human biting rate. This tells us how many bites a single person actually receives in a day. To find this, we need to combine the insect's behavioral preference with its population density and its biological digestion speed. Let us look at how we quantify the feeding frequency next. Before we can calculate the total bites on a human, we must understand the baseline biting activity of the insect. This is represented by the daily feeding frequency formula. Conceptually, this is simply the inverse of the insect's feeding cycle. The symbol F represents this daily feeding frequency. If a mosquito takes a blood meal every 4 days, we divide 1 by 4 to get 0.25. This means it takes 1/4 of a blood meal per day. Understanding this biological clock is essential. If the temperature rises and the mosquito digests blood faster, the feeding cycle shortens. This increases the feeding frequency, which directly increases the speed of disease spread in a community. With our feeding frequency understood, we can now calculate the ultimate risk factor. This formula beautifully combines population size, biological digestion speed, and behavioral preference into one equation. The symbol B represents the total number of human bites received per person per day. The symbol M is the mosquito density per person. The symbol F is the feeding frequency we just discussed. Finally, HBI is the proportion of bites that actually target humans. If the mosquito density M increases, the total bites B will increase proportionally. This equation shows exactly why controlling mosquito populations and changing their biting habits are both critical for saving lives. Let us apply this comprehensive formula to a real village scenario. We are given a mosquito density of 2,000 mosquitoes per person. They feed every 4 days, giving a frequency of 0.25. Their human blood index is 0.60. We want to find the total human bites per person per day. We multiply 2,000 by 0.25, which gives us 500 total bites per person from all animals combined. We then multiply that 500 by 0.60 to filter out the animal bites. The final answer is 300. This means a single person receives 300 mosquito bites every single day, indicating an incredibly high-risk environment requiring immediate emergency intervention. Having calculated the severe risks, let us visualize how these elements connect in the host-vector interaction cycle. On the screen, you see 3 main components forming a triangle. At the top left is the vertebrate host, representing humans or animals. At the top right is the insect vector, our mosquito. At the bottom is the pathogen, the actual disease-causing agent. Notice the arrows connecting them. When the vector takes a blood meal, it uptakes the pathogen from an infected host. The arrow returning to the host represents the biting behavior and host preference, governed by the human blood index we calculated earlier. This diagram shows that the vector is the critical bridge. If we break these arrows, the disease cannot spread. We have covered a lot of ground today, so let us summarize the most important ideas. First, the host-vector relationship is the fundamental bridge that allows vector-borne diseases to exist. Second, behavioral traits like anthropophily, preferring humans, and endophagic behavior, biting indoors, are what make certain insects so dangerous. Third, we learned how to use the human blood index to turn unpredictable insect behavior into clear, actionable mathematical data. Finally, by combining population density, feeding frequency, and host preference, public health experts can accurately measure risk and deploy life-saving interventions. You now have a solid understanding of how insects and diseases interact with human populations.